Why do cells really need to divide? And how exactly do cells divide? In part 2 of Genetics 101, we're going to examine the process of cell division in both mitosis and meiosis. So if you need help in this topic, keep watching. So what exactly is cell division? So cell division involves one parent cell dividing into two or more daughter cells. So we have this one parent cell dividing into these two daughter cells, and you can see the chromosomes highlighted in blue. Now, why do cells need to divide? So the purpose of cell division is really for the cells to be able to multiply, for growth, for repair, and reproduction. So those are the three main reasons behind cell division. And in order for cells to divide, the chromosomes within the cells need to replicate first. They need to make copies of themselves before the chromosomes, the information can be passed on from the parent cell to the daughter cell. So this is occurring within the nucleus, and the chromosomes need to make copies of themselves before they can be passed on to the daughter cells. So replication of chromosomes need to occur. So the genetic information, so all the genes, all that can be passed on from the parent cell to the daughter cells. So there are two types of cell division you need to know about. You need to understand the difference between mitosis and meiosis. So in this video, I'm going to break down the stages of each process so you understand how the chromosomes behave during cell division for each case. So let's begin with the process of mitosis. So as you can see here, before the actual process of mitosis occurs, we have this first stage known as interphase. And this is where the chromosomes are pretty much not as visible under the microscope as you would see them later on. So they're existing as very thin strands, and they have not replicated as yet. So during this phase, replication of the chromosomes, when they make copies of themselves, that would occur in this preliminary phase leading up to the actual beginning of mitosis. So the next phase, prophase, as you can see, the chromosomes are now visible and they're existing as double-stranded chromosomes. So they each have two sister chromatids connected in the middle by a centromere. And the centromere is going to play an important role as the chromosomes are going to be moving throughout the cell during the process. So here we can see in prophase, the chromosomes have thickened, they are more visible now that they have replicated. Now the second step, metaphase, this is when you're going to have the chromosomes lining up along the middle of the equator, or the middle of the cell, and they're lining up in preparation for the next stage. And you can see that there are some dotted lines here representing what is known as spindle fibers. And these spindle fibers are going to be responsible for allowing the movement of the chromosomes to opposite ends of the cells. So at the next phase, anaphase, you can clearly see now that the chromosomes which were once held intact by the centromere, they have now split. Each of them has split, and we have half going up, half of them going down. So they're now existing as individual sister chromatids going to opposite ends of the cell. So they're moving away from each other, they're separating from each other. So that's what happens at anaphase. And as you can see, the process of cleaving, so you can see that one cell is now starting to cleave in the middle in preparation for the actual cell dividing. So at the end of mitosis, telophase, is when you have now the sister chromatids have arrived at opposite ends. And you're now going to see cleaving fully happening here to divide into two separate cells. So at the end of mitosis, we have two diploid daughter cells. So we started off with one diploid parent cell. So remember, diploid means having the full set of chromosomes, usually represented by 2N. So we start off with a parent cell that is diploid, and we end off with two diploid daughter cells. So that is basically what you need to know about the process of mitosis. So let's summarize the key features of mitosis. So at the start of mitosis, you begin with one diploid parent cell. and we're trying to maintain the diploid chromosome number throughout the cell division. So there's not going to be any crossing over of chromosomes, so therefore there's not going to be any genetic variation. So at the end, you're going to have all the cells being produced. They're going to be genetically identical to each other and the parent cell. So at the end of mitosis, you have two diploid daughter cells. Okay, so now we have an idea of the process of mitosis and the key features. Let's look at the purposes of mitosis. So one of the main purposes of mitosis is for growth. Now let's look at this first example, embryo development. So this is the process of development from the zygote after fertilization occurs. So when the sperm fertilizes the egg, you have the zygote being formed. And that fertilized egg then undergoes a process of a series of mitotic divisions. So we have lots of mitosis divisions occurring in this fertilized egg, the zygote, to produce the actual embryo. Alright, so another example would be obviously growth from childhood to adulthood. So that embryo will eventually grow till it forms a fetus, which is a young baby, and then after birth, obviously the baby grows and develops into a child, then an adolescent, and then to the adult. So in that process of growth, obviously cells need to be dividing. All the tissues and organs, they need to be increasing in size as the individual is getting taller and wider. So that is an example of mitosis in growth from childhood to adulthood. And then obviously it occurs in plant growth as well. So that is how the roots and the shoots would grow. So they usually have certain parts of the roots and shoots known as the meristems where mitosis would occur, and that would allow the roots to get longer and the stems to get taller and wider, and obviously the leaves to get bigger. So these are three examples of mitosis in growth. Now mitosis is also important in the actual formation and repair of tissues. So cells divide by mitosis to produce specialized cells. So these are cells with specific functions. So these cells will obviously form new tissues. They will replace old, damaged, or dead ones. So it's important for mitosis to be allowed to actually produce and make these cells in the body. Now, a common example, so if you think of it this way, if you get a cut and your skin is wounded, your skin needs to be able to repair itself. You want to be able to have skin cells coming back and replacing those old damaged ones. And think of the whole concept of mitosis producing identical cells. You would expect and you would hope that the skin that is coming back to replace the old damaged cells should be somewhat identical or similar to the old ones. So that those new skin cells should definitely have a similar appearance to the old skin cells. So generally speaking, mitosis is needed to produce all these different specialized cells, non-reproductive cells that is, within the body, and is extremely important for the repair of these same cells, these tissues, if they get worn out or damaged or die. Now, the last purpose of mitosis would be in asexual reproduction. So asexual reproduction, that is when you have a single parent producing identical offspring without the formation of gametes. So in asexual reproduction, so let's look at this first example. The bryophyllum plants is a good example to look at. So mitosis would occur at the meristems along the leaf margins. So as I'm pointing here, you can see this is the margin of the leaf, and you're seeing little plantlets appearing on the margins. So these leaves, the leaves from the parent plant, produce these tiny little plantlets that will eventually fall off and grow in the surrounding area. And all of these plantlets are going to have the same genes. They're going to be identical to the parent plant. So that is an example of asexual reproduction there. And then similarly with the strawberry plants, now instead of the new plants growing along the margins of the leaves, we have buds on the runners. Those are the horizontal stems that you're seeing there. So the buds are produced by mitosis, and then you have new strawberry plants being formed from these buds on the runners of the parent plant. So those are just two common examples of asexual reproduction and how mitosis plays a role in that process. Okay, let's go on to look at the process of meiosis. Now, meiosis tends to be the one that could be a little tricky and a little more challenging. So the first thing you need to understand about meiosis is that there are 2 rounds of cell division, unlike mitosis, which only has 1 round. So here we're looking at the first round of meiosis, and we're going to call it meiosis 1. So the first phase of meiosis, prophase 1, you're seeing that the chromosomes, and they're homologous pairs of chromosomes as you're seeing them arranged in the nucleus of your cell. So they're pairing up. So you should remember that a homologous pair of chromosomes includes 1 chromosome from the mother, 1 chromosome from the father, and they have the same sequence of genes along the length of the chromosome. Now, as you can see here, an important event is happening in meiosis at this stage. The chromosomes are actually intertwining and connecting through the process of crossing over. So this is where you're going to have an exchange of genetic material, and I'll go into that a little more shortly. Now, at this stage in prophase, so we have the homologous chromosomes, they're pairing up, crossing over is occurring. So the next stage now, metaphase, we're going to have these homologous chromosomes lining up, and you can clearly see the results of the exchange during crossing over. So you have a little piece of the mother's chromosome with a little piece of the father's chromosome. So they're lining up now at the middle of the cell, preparing for separation. Now in meiosis 1, so let's go over to anaphase 1 now, you're going to see that the actual chromosomes are not going to actually separate in terms of splitting into individual sister chromatids at this stage. So the centromeres are still intact. So you're going to have 2 chromosomes going up, 2 chromosomes going down, and they're still connected to each other. They have not actually split and separated into the individual sister chromatids. So this is what's happening at anaphase 1, and you can clearly see the difference there when you compare it to mitosis. So there's no splitting into separate sister chromatids as yet. The chromosomes are still intact. Now at telophase 1, we're going to have the cleaving occurring, 2 cells being produced, and you can see clearly that they now have 2 chromosomes in each cell. So this is important in meiosis. As you can see clearly, the chromosome number has been halved, because at the beginning we had 4 chromosomes. If you look carefully, there are 4 chromosomes, they're intertwined and everything, but there are 4 chromosomes shown in the parent cell, and now we have 2 chromosomes shown in each daughter cell. So that is the end result of meiosis 1. Okay, let's summarize these main events of meiosis 1. So first of all, we started off with 1 diploid parent cell, represented by 2N. Now the homologous chromosomes pair up, and we have the crossing over, also known as synapsis. So that is where you have the exchange of gene segments on the chromosomes from the mother's chromosome, from the father's chromosome. So this is also known as recombination. So you're going to have a recombination of genes from the mother and the father's chromosomes. So crossing over is a key event in meiosis 1 that you need to understand. So with this exchange of gene segments, you can clearly see where the genetic variation is going to come in. So the homologous chromosomes are then going to separate. So this is where they're still intact with the centromere connecting the 2 strands together, and they're going to move to opposite ends of the cell. So this is where the halving occurs. So we start off with the full set of chromosomes in the parent cell. So at this point now, this is where halving is going to occur, and the cells are going to start to become haploid. So the cell cleaves and divides after the chromosomes will reach the end. So finally, at the end of meiosis 1, we have 2 haploid daughter cells, denoted by N. So we now have half the chromosome number for each of these 2 daughter cells. So let's look a little closely at what happens during crossing over, so you get a better idea of what's really occurring. So we have our homologous pairs of chromosomes here. So 1 homologous pair of chromosomes here, and you can see that there's genes represented by the letters A, B, C. So we have 1 from the father, 1 chromosome from the mother. So let's say the father's chromosomes represented by the blue, the mother's chromosomes represented by the red. So you can see that the sequence of genes are the same along both chromosomes. Now during crossing over or synapsis, you can see that piece of the father's chromosome is intertwining with piece of the mother's chromosome. So therefore, at the end of crossing over, we have this exchange of genetic information, and this is where genetic variation actually originates from. So you can see that we have a piece of the mother's chromosome attached to the father's chromosome, a piece of the father's chromosome attached to the mother's chromosome. So this is where you get those differences coming about. Okay, let's move on now to meiosis 2. So this is round 2 of meiosis. So we're picking up from where we left off with the 2 haploid daughter cells being produced. So this now goes into meiosis 2. So we're starting off here at prophase 2. So the 2 daughter cells, they contain 2 chromosomes each, and you can see the evidence of crossing over. So now from here on, the process of meiosis 2 is going to be quite similar to mitosis. So where you're now going to see in metaphase 2, the chromosomes are aligned at the middle of the cell in each case, and then from there we're going to have the splitting of the chromosomes into these separate sister chromatids. So that's what's happening here at anaphase 2. So we have the chromatids, the sister chromatids splitting and separating and moving to opposite ends of the cell. So at the end of meiosis 2 now we have 4 daughter cells carrying the 2 chromosomes. So that completes the process of meiosis. So let's sum up meiosis 2. So we had 2 haploid parent cells to begin with. So those came from meiosis 1. These sister chromatids, which are each strand of the chromosome, separates and moves to opposite ends of the cell. So this is similar to the process of mitosis. So this is similar to what happens during anaphase in mitosis. Now each cell will divide after the sister chromatids reach the end, and each daughter cell is now genetically different from each other and the parent cell. So it's a combination of the crossing over and the halving of the chromosome number that brings about these genetic differences. So at the end of meiosis 2 now we have 4 haploid daughter cells denoted by N. So 4 haploid daughter cells, all of them are going to be different from each other. So we have genetic variation produced. Alright, so now let's look at the purpose of meiosis, and that is in sexual reproduction. So meiosis is really meant to produce gametes, the sperm and the egg. So in humans and animals, you would find meiosis occurring within the testes of the male and the ovaries of the females. So the testes is where the sperm will be made in the males, and the ovaries make the eggs. So these are the gametes. So that is in humans and animals. Now in plants, flowering plants that is, the pollen, which is produced by the anther, that's the male part of the flower, the pollen carries the male gametes. And then the ovule, which is part of the ovary, the female part of the flower, would carry the eggs. So you see exactly where meiosis occurs within animals, humans, plants, and obviously because the meiosis process is meant to produce gametes, you can see that meiosis is extremely important in the whole process of sexual reproduction. So that is really the main purpose of meiosis, to produce the gametes which are involved in sexual reproduction. Alright, so let's just summarize the differences between mitosis and meiosis using these 2 diagrams here. So clearly in mitosis, we start off with 1 parent cell that is diploid, and we end off with 2 daughter cells that are also diploid. So it's just 1 round of cell division. In meiosis, we start off with a diploid parent cell, and we're having 2 rounds of cell division, and you're going to end off with 4 daughter cells that are haploid. And in a diagram like this, you should be able to identify which of them is mitosis, which is meiosis, by examining the number of cells and the number of chromosomes in the daughter cells as compared to the parent cell. So clearly this has to be mitosis because the parent cell, as you can see here, it has 4 chromosomes. If you look at the daughter cell, it also has each of them having 4 chromosomes as well. So clearly these cells are identical to each other, same chromosome number. So that's where we have the diploid number being maintained. Now with meiosis, We started off with 4 chromosomes. Remember, it's a diploid parent cell. But at the end, we have 2 chromosomes in each of the 4 daughter cells. So each of those cells would have half the chromosome number as the parent cell. So clearly that has to be meiosis. So those are the key things to look for when you're trying to determine which cell division is mitosis and which one is meiosis. So this table here just gives you a comparison of the 2 types of cell division and the key features that you should know for both of them. So in the next video, we're going to go on to look at inheritance of traits. So stay tuned for part 3 of Genetics 101. If you found this video helpful, feel free to subscribe, like, and share. And don't forget to hit that notification bell.